Good morning, everybody. My name is Catriona Dowling, and I'm just finished the second year of my PhD, and the title of my project is Targeting Protein Kinase C in Colorectal Cancer. So firstly, I just want to give you a brief overview of what colorectal cancer is and how exactly it develops. So in the colon you have normal mucosa, and then this normal mucosa undergoes an accumulation of genetic alterations that results in a benign mass of cells known as a polyp. This polyp will then undergo another series of genetic alterations, and then the tissue becomes malignant, and this is when you have colorectal cancer. So our question is, if we could just understand why this accumulation of genetic changes happens, it would help us in the early detection of colorectal cancer. So this is where our protein comes in, called Protein Kinase C. So PKCs are a group of proteins in the cell, and they're a very important group of proteins. They're involved in a number of very important pathways in the cell, such as in pathways in proliferation and migration and invasion. There are 9 different types of these PKCs, so 9 different isoforms, and they're divided into 3 different families. So you have the conventional, the novel, and the atypical PKCs. These PKCs they're just divided depending on what activates them. So the PKCs are differentially expressed depending on the type of tissue that you actually look at in the body. So our hypothesis is that as the tissue goes from this normal phenotype into the transformed malignant phenotype, there's a change in the activity and the location of these PKCs. So also we want to take into consideration, we know it's very important to take into consideration the stromal environment around these colon cancer cells, trying to understand what's going on there as well. So we began by developing a series of models. So our first model was we had these rounded cells here are your colon cancer cells, and the long elongated cells are your fibroblast cells. So we grew colon cancer cells in the presence of fibroblast cells in a 2-dimensional matrix. We then went further with this and developed another model in which we grew colon cancer cells in a 3-dimensional spheroids. So when you extract these spheroids, I stain them here just so you can see clearly what they look like. So these are colon cancer cells growing in a spheroid. And then we laid down fibroblasts around these spheroids to look and see, well, what effect is having on the development of these spheroids. So now I wanted to take this another step further and see, well, okay, well, what actual effect is having on the rate of proliferation and invasion on the cancer cell. So I came up with a method to look at this. So firstly what I did is I took fibroblast cells and I put them into 2-dimensional culture. I then 24 hours later extracted the media from the fibroblast cells. Then I put this media onto this system that we have in our lab at the moment, and it's called the Excelgen system. So the Excelgen system is a real-time monitoring system, so you can monitor cells over a long period of time. So on the system there's gold electrodes, and there's a series of gold electrodes, so you can plate your cancer cells on these gold electrodes, and then you can monitor the rate at which these cells are growing. So we placed our cells onto these plates, and then I put the media on top of these plates, which allowed me to actually see, well, how fast are these cells growing when you put the fibroblast media on them, and you get a readout like this. This gave us some very interesting results. So the first result is that we saw is, yes, when you put the fibroblast media from on top of these cancer cells, there's an increase, you can see here in the green line, so this is cells growing in the presence of fibroblast media. So there's an increase in the proliferation of these cells, and we're able to monitor this over 24 hours. I then wanted to see, well, okay, it is definitely affecting the proliferation of these cells, but another important mechanism is the invasion, and is it affecting the invasion of these cells? So we came up with another method here where we use Matrix, which creates a layer that the cells have to invade through. And we forced the cells to invade through this, and we found, interestingly, that when there's fibroblast media present, the cells will actually invade at a much faster rate. So we also showed this for migration and adherence. We showed that the rate of this was also faster, and we published it earlier this year in Bioscience Reports. And we're now working with Professor Calvin Coffey to use patient-specific media and actually see, well, does this media also have the same effect? So then I wanted to go back and look and see, well, is PKC expression actually affecting the formation of the cancer? So I used real-time PCR here to look at this. And what we did was, tissue was taken from the cancer of patients and matched normal tissue from the same patient was taken. So we had cancer tissue and we had normal tissue from that exact patient. We then extracted RNA from this, did cDNA synthesis, and did a real-time PCR reaction, which took us right through to gene expression. So we were now able to see, in our 9 different PKCs, is there a change when you look at the cancer tissue of a patient in comparison to the normal tissue of that patient? So when we were designing this experiment, we also adhered very tightly to the MICI guidelines to ensure accuracy of results and that there was no problem with our RNA. So for each experiment we did, we tested the quality of that RNA for each patient. We tested the integrity of the RNA to ensure there was no degradation of RNA. And we also did a test where we ran 9 housekeeper genes And then we picked the top 3. So for every experiment, we used 3 housekeeper genes to normalise our experiment. So this ensures accuracy of the results. So we found very interesting results here. So this graph represents the fold change. So this represents the change in the expression of PKCs in the cancer of patients. So this is the change in the cancer tissue. And we were also able to correlate this with the tumour stage. So interesting, we found this gene called PRKCB. So PRKCB is a gene that codes for the protein PKC beta. So we found that there is a distinct downregulation of this protein, of this gene, in all of our cancer patients. So this is a cohort of 24 patients. We also found another gene, which is PRKCG, and this gene codes for the protein PKC gamma. And very interesting here, we found that there was an upregulation of PKC gamma. So gamma is a protein that is only traditionally ever found in the CNS. So when we were looking at the normal tissue of patients, we weren't seeing this protein, but then when we were looking at the cancer tissue of the same patient, there was a switch back on of this protein. So I wanted to confirm these results at a protein level and with a larger cohort of patients. So far I was showing you the change at a gene level, but I wanted to make sure this is actually translated and there's a change at the protein level also. And we wanted to use a large cohort of patients. So I spent a month working with Professor Jacinta O'Sullivan in Trinity College, where we used tissue microarrays to validate our results at a protein level. So just briefly, how these microarrays are created is that you'll have blocks of tissue and cores will be taken, a tiny core from each block will be taken. These cores will then be placed onto another block, and then these blocks are sliced, and then you end up with loads of glass slides which have your cores on the glass slides. You can then take these glass slides and you can stain them for whatever protein you wanted. So I stain these for PKC beta, so that protein, and the protein PKC gamma. So then when you look at these, you can analyse them and look and see, well, is there a change in this protein across your patients? So this analysis is ongoing at the moment, and I'm analysing them as well as a student in Professor O'Sullivan's lab in Dublin, and then the results will be correlated together, and we will do statistical analysis to see is there any result. So when you stain for your protein, you get this brown colour. So wherever you're seeing the brown colour here, that's where there's a protein present. So you can see that it's much more faint here. So this core here is a core taken from the normal tissue of a patient. It's because you can see the cells are nicely differentiated. Here is a core taken from a cancer tissue of a patient, so you're losing differentiation of your cells. This brown colour you're seeing here is actually for that staining that I did for PKC beta. So preliminary results that we have done have shown that yes, what we were seeing at a gene level is being translated across to the protein level. So you can see quite a lot of the protein in the cancer of the patient, in the normal tissue of the patient. When you move to the cancer tissue of that patient, there seems to be downregulation of this protein. So future direction for this project is, I want to further correlate the expression of PKC gamma and PKC beta in tumour stage. I then want to try and look closer at the cells and map the actual movement of gamma and beta and see, well, why is this switch important? And then I want to try and block their activity by using a series of inhibitory peptides. I'd just like to acknowledge Dr. Patrick Kiley, Professor Jacinta O'Sullivan, Dr. Tara Dalton, and Professor Calvin Coffey, who provides with the tissue and my funding of the Irish Cancer Society and the Midwestern Foundation. Thank you.
Okay, so my name is Aoife Culliton, and today I'm just going to talk to you about my work on the development of a novel injectable bone cement. So this work is done as part of my PhD in UL with support from an industrial sponsor also. So just to launch straight into it, so there's a number of synthetic bone substitutes on the market at the moment. So we'll just have a quick look at the existing products. My main focus is on calcium phosphate bone substitutes. So many of the products on the market at the moment actually consist of a powder and a liquid which come packaged separately. This powder and the liquid then need to be mixed together manually. They need to be loaded into a delivery device, and then from there then they can be injected into the defect. So the problems with this is obviously that it requires on-site powder liquid mixing, which is going to increase the length of the surgery, which in turn then is going to add increased risk to the patient. So this highlights the need then to develop a bone substitute which does not require on-site mixing. So that brings us to our aim, which is to develop a dual-paste calcium phosphate-based bone substitute, which comes preloaded in the delivery device, and when injected then will result in a self-setting injectable bone cement. So this will require no separate mixing by the surgeon and reduce the time spent by the patient in surgery. So we have this dual barrel syringe here. So on one side we'll have an aqueous paste, which will provide the water required for the reaction, and then we'll have a non-aqueous paste on the other side. So once the syringe is injected, the aqueous paste and non-aqueous paste will be mixed through this static mixing cannula. So once it's from there, then it can be injected directly into the defect, requiring no separate mixing. So just a quick look at the formulation chemistry involved. So we've got our non-aqueous paste components here and our aqueous paste components. So basically what's happening is reaction 1 happens first. So we've got the reaction of monocalcium phosphate monohydrate from the aqueous paste is going to react with tetracalcium phosphate from the non-aqueous paste first of all. So this is going to form dicalcium phosphate dihydrate. So this DCPD is responsible for the initial hardening of the cement. Then reaction 2, the DCPD that's formed is going to react with the tetracalcium phosphate in the non-aqueous paste. And while this reaction is going on, so while reaction 2 is happening, reaction 3 is also going on. So we've got the DCPA here from the aqueous paste also reacting with the tetracalcium phosphate. So both reaction 2 and reaction 3 are going to form HA, hydroxyapatite, which is the end product of our cement. So we had a number of design requirements from our industrial sponsor. So one was for the aqueous paste to have a settling rate of less than 1500 millimetres per year, and also the non-aqueous paste was to be thermally stable up to 50 degrees. And then on the surgical side then, so surgeons' requirements were that the injection force had to be below 80 newtons, and the injection time was to be below 1 minute. Wet field characteristics were to be similar to that of an existing product, Hydroset, on the market, and the compressive strength is to be greater than 6 megapascals. So in order to do that, we had to optimise both the aqueous paste and the non-aqueous paste. So if we look at the aqueous paste first of all, the variables that were considered were the particle size of the 2 calcium phosphates in the aqueous paste and also the PVP quantity. So after optimising these 3 variables, 19 potential aqueous pastes were generated. And similarly with the non-aqueous paste, so after optimising these variables here, 10 potential non-aqueous pastes were generated. So each of the 19 aqueous pastes were combined with each of the 10 potential non-aqueous pastes, resulting in 190 combination pastes. So from these 190 combination pastes, the settling rate, injection force, compressive strength, and the wet field characteristics were tested, as these were our initial design requirements. So the 4 best performing pastes in these regards that met the design requirements were carried on further for further characterization. So these 4 combination pastes then were the effective gamma radiation on these pastes was tested, the thermal stability, they also underwent thermal stability testing, and also XRD of all of these 4 pastes confirmed the presence of HA as an end product, which is our aim. So from these 4 combination pastes, the final paste was chosen by our industrial sponsor. So with this final paste chosen then, there was further characterization carried out of this final paste using a number of techniques. So we used analytical techniques such as X-ray diffraction, FTIR and solid state NMR. And then we also used imaging techniques such as SEM, TEM and micro-CT. So obviously I can't go into detail on the results of all of these, so we'll just focus on a few of these techniques at the moment. It'll load up. Okay, so solid state NMR is one of the techniques that we can use to monitor the formation of HA as a function of time. So here we've got scans, we've got NMR scans as a function of time, so we've got scans from 2 minutes up to 14 days. So this is our 2-minute scan here, so you can see all that we can see here at 2 minutes is our raw materials. So these 2 peaks here are tetracalcium phosphate, and then these 2 peaks here are dicalcium phosphate, which are 2 raw materials. And the MCPM, which is another raw material, is actually gone. So that means reaction 1 that I was talking about earlier on has happened already by 2 minutes. So there's no MCPM left in the cement. So the main area of interest then is this area here in the middle. So we can see our product formation. So this is the growth of HA as a function of time. So we can see our HA forming. So then to look at some of our imaging techniques that we used. So this is just an example of some of the crystal morphologies that we've seen using SEM. Now, the only way to accurately identify these crystals is by isolating one single crystal and analysing it using TEM. So isolating a single crystal at this scale has proven quite difficult. So we have used a customised sample prep technique for TEM, where we've been almost able to isolate a single crystal, but we can see that there's still some overlap, and there's actually some kind of overlapping crystals underneath this one as well. So work on this is ongoing, so we're continuing to try to isolate these crystals and further identification of what we've got going on in here. So then we can use micro-CT as well. So micro-CT basically just takes thousands of cross-sectional images of your sample, stitches them all together, and you can recreate a 3D model of your sample. And this allows us to see the internal microstructure of our set cement. And we can also use micro-CT to monitor porosity evolution as a function of time. So this is a 3D reconstruction of the pore network within the sample. So we can see at 1 hour, the sample is highly porous compared to 14 days. So if you actually look at the large particles here, so this are the large pores. So this large pore seems to remain unchanged after time. However, a lot of the small pores seem to have diminished. So what we think is happening is the HA is actually growing into the area where the small pores were, and that HA then is going to contribute to the overall strength of the material. So just to conclude, a stable injectable product has been developed with an injection force of 17 newtons and a compressive strength of up to 11 megapascals. Although we couldn't go into the results of them all, XRD, solid-state NMR and FTR analysis has shown that the chemistry of this product is very similar to the chemistry of existing products on the market. Understanding the porosity evolution may allow the cement to be tailored further, so in terms of drug incorporation and drug elution rate into the cement, if we decided to go down that route. And so ongoing work, obviously, there's further characterization required of the phase evolution, tailoring the porosity to enhance osteointegration, and ongoing work using TEM for phase identification. So I'd like to acknowledge the following people, and thank you all for listening.
Good morning, my name is Elena, and today I'll be talking to you about a simple in-vitro fluid flow bioreactor for fluid flow stimulation to enhance osteogenic commitment of human mesenchymal stem cells. So bone structure adapts to meet the demands of its local mechanical environment, as seen by enhanced bone formation in a tennis player serving hand, or decreased in bone mass due to microgravity. Physical loading induces fluid flow within the bone, which is known to enhance bone formation. Stem cells present within the bone marrow can differentiate into bone cells under loading, but the optimal mechanical stimulus and the mechanism behind it is unknown. In vitro bioreactors enable to study how physical loading directs stem cell differentiation, and this can be used to develop tailored physiotherapy regimes for bone regeneration, as well as optimized strategies for bone engineering tissue grafts. Current platform for fluid stimulation of cells is the parallel plate flow chamber, which is the gold standard. It presents a narrow channel where fluid is pumped through, inducing a shear force over the cells, stimulating them. And our lab has validated this system to enhance osteogenic potential of human mesenchymal stem cells, as seen by upregulation of osteogenic markers. However, this system has some limitations. It is actually quite difficult to use and expensive. It poses high risk for contamination, and it is not available commercially. Therefore, my aim is to develop a simple, user-friendly vitro bioreactor system to enhance osteogenic potential of human mesenchymal stem cells using fluid flow for tissue engineering strategies, and as a model system to study how cells react to fluid-induced shear forces. So the design criteria for my bioreactor is that the mechanical stimulus should be characterized so that I know what kind of forces apply to my cells. It should enable long-term culture, easy to use, cheap, and it should enable the use of existing lab equipment. Rocking platform is a common piece of equipment that has been used in the past to stimulate cells with fluid flow. So the shearing motion induced by the flow is that when you culture cells in a dish, similar like that, during the tilt motion, the fluid travels in the direction of the tilt, inducing a shearing force over the cells, like this, stimulating them. And this shearing motion can be defined as a function of the tilt angle and frequency. However, the system is not popular due to the fact there is an uncertainty in the mechanical stimulus applied to the cells. Therefore, characterizing what kind of shear stress ranges and magnitudes apply to the cell monolayer is quite important for experimental design. Zhu recently developed a mathematical model that describes how shear stress is distributed and the magnitude of a cell monolayer during a rocket motion, and it is a function of a maximum tilt angle, the fluid height in the dish due to how much volume you add, dimensions of the dish, the frequency, and the critical tilt angle to ensure that even during the tilt motion, the cells are covered with fluid to ensure the cells won't die. So I wrote a custom computer program that computes quite easily all these parameters from this horrible-looking equation so that anybody can apply it. What I'm interested in is in mimicking bone biomechanical environment to stimulate cells with fluid flows to induce an osteogenic response. So the rocking motion induces a special temporal oscillatory fluid flow, and I characterized for a 6-O plate to induce the maximum shear stress, and this can be achieved with 0.2 milliliters of volume, 2 degrees tilt angle, and 0.82 Hz frequency. And this is how the shear stress looks and the distribution. So for example, if you take the middle of the well, the shear stress during one tilt motion goes from 0.1 Pascal down to 0 to 0.1, and then opposite direction goes from 0.1 again to 0 to 0.1. So now I have characterized that my shear stress ranges from 0 to 0.12 Pascal, which is so physiological magnitude, and the distribution is non-uniform, which is actually what happens in the bone. So I'm now interested in applying this model system and see whether I can induce stem cells differentiating into bone cells. So biologically validating the system, human mesenchymal stem cells are cultured for 2 days, subsequently divided into 4 groups. My negative control, which is growth medium, is static culture. My no-flow control, which is static culture with minimal osteogenic supplementary growth medium. My flow group, which were cells grown in minimal osteogenic supplementary medium and stimulated for 4 hours a day for 3 consecutive days. And my biochemically induced osteogenesis as a positive control. The use of no-flow control will delineate specifically the effect of flow independent of osteogenic supplements, as they are used to support the osteogenic process. So immediately post-flow, the MRI expression of osteogenic factors was quantified. Additionally, the cells were grown for 21 days to identify where I have functioning osteoblasts depositing bone matrix using alizarin red staining and the calcium concentration in the matrix deposited. So immediately post-flow, an increased osteogenic response is seen in the flow group. Specifically, COX-2, which is an enzyme involved in bone formation, is significantly upregulated in comparison to no flow, and similar to my biochemically induced osteogenesis. Additionally, osteopontin and BMP2 are 2 bone matrix proteins, and they are significantly upregulated in comparison to no flow, and much higher than the biochemically induced osteogenesis. 21 days later, enhanced mineralization is in the flow group in comparison to no flow, and quantifying the calcium content of that matrix is in there's a 2-fold increase in calcium in comparison to no flow, therefore validating my model that even at physiological magnitude of mechanical stimulation, I have stem cells differentiating into bone cells and actually reducing bone matrix. So in conclusion, the rocking platform is presented as a novel bioreactor system for fluid flow stimulation, where the design criteria was met that mechanical stimulus was characterized, it enabled long-term culture, it was easy to use, cheap, and the equipment already existed in the lab. It poses some limitation that the shear stress range magnitude was physiological and the distribution non-homogeneous, but even under these circumstances, I was able to show that the stem cells differentiated into bone cells. Moreover, this system can be applied for any other tissue engineering applications and mechanobiology studies. I would like to thank you, my lab, and the supporting funding bodies. Thank you.
I'm Marie Keays, and today I'm going to present on the development of a microfluidic cell culture in smooch. So I'm working in the Stokes Institute, where I work in a multidisciplinary team, and our aim is to make biological research more effective and more efficient. And our focus in our group is on breast cancer and why it metastasizes. So when we take a sample from a patient, what we can get is genetic information. But what we would like to be able to do is take a sample from a patient, yes, get the genetic information, but also test hundreds of different drugs against these samples in a low-cost way and a high-throughput way so we can get the most amount of data for the smallest sample possible. And how I'm going to do this is using microfluidics and cell culture techniques. So current cell culture techniques is where you take cells from the body and grow them outside the body in either a dish or a flask. The problem with this is that each dish is an individual experiment. So if you want to do hundreds of experiments, you need hundreds of these. And this just isn't cost or time effective because each of these have to be manipulated and manually monitored. And most of this research is done in 2 dimensions. So 3-dimensional culture has been shown to be more biologically relevant and mimics more closely how cells react in the body. So what microfluidics allows us to do is look at cells in their own length scale. So cells here, the red blood cell and bacterial cell, how they're cultured at the moment in flasks are orders of magnitude bigger than the cells themselves. So microfluidics under 1 millimetre allows us to monitor in their own length scale the time it takes for the cells to divide, and also we can monitor how 3D culture develops. So the type of microfluidics that I'm going to use is droplet microfluidics. So here we have a droplet that contains the cells and the culture media. It's about 500 nanoliters in volume and 1 millimetre in length. It's contained within this tubing, which is gas permeable, so allows the gas in and out of the culture. And the carrier fluid that flows these droplets through the tubing is an oil. This oil also creates a barrier around the droplets, so we essentially get no contamination within the system. We can also tailor the environment depending on the cell type. So if the cell is an adherent cell, we can add an adherence material such as a gel matrix, or we can keep it in suspension for a suspension cell. And you can see here in 1 centimetre we have around 10 experiments, so we've already reduced the size of these experiments. So the system that I have created to develop these droplets is here, where we have a syringe pump which pulls the flow through this tubing. So this is mainly pulling oil through the tubing. And when you want to create the droplets, you use your dipping stage, which is programmed. You push into the sample, which is your cells and your media, and you pull back out again. This creates the droplets and allows them to move through the tubing, past the microscope and into the incubation region. We can monitor these cells at any time point during their incubation through the microscope, but we've also developed a system for measuring optical density of these droplets. So we don't have to lose our sample to a plate reader. So here's the system, what it actually looks like: the pump, the heater plate, and the sample and the dipping stage that moves in and out of the sample. So the advantages of this type of culture is that each droplet is a unique experiment, meaning that we have a 500 nanometer droplet that contains the cells and whatever you want to test it against. We can control the volume of these reagents. So we have, if you take for example 1 typical 10 milliliter flask is equivalent to 20,000 of these experiments. We've made this a high throughput system, and we made it cost effective by reducing it in this scale. And we're also able to monitor these droplets without taking them out of the system. So it allows for real-time processing. So to test this system, we looked at MCF7 breast cancer cells. And you'll see here initially when we put these cells into the droplet, which is 1 millimeter in length, that all these little dots here are cells spread throughout the droplet. After 24 hours, you can see that these droplets have accumulated together to form a 3-dimensional structure. And after 48 hours, this structure has got bigger. We've also looked at this system through confocal, and we've been able to see that this this looks like a 3-dimensional spheroid. So ideally what we would like to be able to do is take a sample from a patient, grow it in this way, form these spheroids, and get a drug response in this system. What we also looked at was bacteria culture. So with bacterial resistance becoming more important, we want to see how bacteria would actually react in these droplets. So we took E. coli culture and we cultured it in its typical way in an orbital shaker flask and compared this to a 600 nanoliter droplet. You'll see here that we get a response from both, but they both grow. And after 10 hours, you can see that the droplet culture has actually grown at a faster rate than the typical control culture. And the reason we think for this is the higher surface volume ratio allows more diffusion in and out to the culture itself. What it really tells us is that we can take bacteria, grow it in the system, and get a response within 10 hours. So what I have been able to do is create a system to create these droplets, being able to grow bacterial and mammalian cells, and create three-dimensional structures. So this has potential to be introduced into any lab, from your research lab up to your big pharma, just due to the cost savings. What's next for this system is to add a liquid bridge. So this mixing system has been used for other Stokes technology, so it's a proven technology. What it allows us to do is have one droplet that contains cells, one droplet that contains a drug that you want to test it against. These come together and join as one droplet. What this allows us to do is have any drug you want to test against any droplet media or any droplet culture. Being able to take a sample from a patient in such a small sample size allows us to create thousands of experiments and bring us closer to personalised medicine. So being able to do this with bacterial and mammalian cells means that this is one system that has endless possibilities for cells. I'd just like to acknowledge my supervisors and thank you for your attention.
Good morning. So I'm in the final stages of my PhD at the UL Graduate Entry Medical School, and my thesis was conducted as part of a 3-year Health Research Board funded project which was led by Professor Walter Culliton and which aimed to develop an early intervention for mental health and substance use problems that was evidence-based, feasible and acceptable to young people. So for the purposes of this presentation this morning, I'm going to discuss some of the findings from a cross-sectional study with GPs to assess their attitudes towards treating youth mental health problems. So just to give you an overview of the literature, mental and substance use disorders contribute the largest disease burden in young people, with 75% first emerging between the ages of 15 and 25. And McGarry and colleagues suggested that providing early intervention for treating mental health is a best buy in terms of being more cost effective and efficient than treating adults with mental disorders. However, GP detection rates are quite low for detecting mental health problems, and qualitative studies with GPs have highlighted the following barriers when trying to address youth mental health problems in young people, such as fear of over-medicalising young lives and resource issues around lack of available GPs and inadequate training in youth mental health. However, previous research has also reported the benefits of training GPs in screening and treating youth mental health problems, and significant gains were noted in GP recognition of psychological distress, depression and suicidal ideation, and also significant increases were noted in clinical skills, knowledge and GP confidence to address youth mental health problems. So our mental health and primary care research group at UL has been engaged in a programme of research to support the GP in addressing youth mental health problems. Work to date has included a discussion paper which outlines the importance of multifaceted interventions for enhancing the GP role to address youth mental health problems. Cross-sectional studies in the Midwest and Dublin South inner city have identified youth mental health as a major problem in economically deprived areas. Qualitative studies with healthcare professionals and young people from primary care, secondary care and community agencies in Dublin South inner city and Limerick City identified the key enablers and barriers to screening for youth mental health problems. Key areas included resource issues, the need to promote mental health awareness and also further training for GPs in this area. A Delphi study with an expert stakeholder panel of healthcare professionals identified 5 core areas for implementing guidelines to address youth mental health problems. Such areas included prevention, assessment, treatment, interaction and ongoing care. So based on our previous work, we developed a GP masterclass which was held in UL back in April to improve how GPs address mental health problems in their practice. So the main aim of the current study then was to examine the role of the GP in providing early intervention for mental and substance use problems. Additional objectives were to examine current factors associated with identification and treatment, and also to look at factors associated with screening, brief intervention and referral. A national random sample of GPs were identified from the Irish Medical Directory. 183 GPs responded, so we were provided with a 50% response rate. Data analysis included descriptive analysis using independent sample t-tests, chi-square tests, and logistic regression was used to determine the main predictors associated with screening and referral and brief interventions. The questionnaire design was informed by international studies looking at GP attitudes to addressing youth mental health problems, previous work by the Mental Health and Primary Care Research Group, and the instrument was reviewed by the Project Steering Group, research colleagues and CSTAR at UL. Key variables included in the questionnaire included demographic variables, strategies adopted by GPs to address youth mental health, current level of training and previous attitudes towards previous training received in youth mental health, barriers and enablers to effective care of mental health problems, and GP attitudes towards working with youth mental health, and also their attitudes towards incorporating brief interventions in practice. So the study sample included mainly GPs in the older 50 plus age range, with only 5% in the 35 age group. 82 males and 79 females took part, and the mean number of years post-GP training was 22. The mean number of doctors at a practice was 3, and the majority of GPs worked in mixed practices, treating both GMS eligible and private patients. On multivariate analysis, factors associated with associated with not screening for mental and substance use disorders included more than 20 years training as a GP, and the lack of counselling services available at the practice. GPs who identified education and training as a barrier were also less likely to screen, and confidence to treat substance use disorders remained as a predictor of screening for substance use disorders. Counselling services delivered at the practice was associated with using CBT to treat mental disorders. GPs in the 50 plus age group were more likely to use counselling for treating mental and substance use disorders, and also GPs who were satisfied with their training in child and adolescent mental health were more likely to screen for mental disorders, and GPs who were involved in CME were more likely to use counselling services for substance use disorders. Interestingly, GPs with more years training were less likely to use web-based interventions. So GPs based in mixed practices were more likely to refer moderate cases of mental and substance use disorders to specialist care. Counselling services delivered at the practice was also, or the lack of counselling services delivered at the practice was also associated with non-referral, and those who were also identified their lack of education and training in youth mental health were also likely to refer mental and substance use disorders to specialist care. So when asked to identify the key barriers to addressing mental and substance use disorders, the key areas included attitude of the patient and our family members, lack of specialist staff in the practice, lack of time and poor service availability. And when asked to identify key interventions to enhance their role to address youth mental health problems, the key areas were access to services, particularly psychological services, appropriate time and space to explore youth issues, clear definitions of which interventions can be addressed in primary care, and access to a youth worker. So to conclude then, given the considerable contact young people have with primary care, it's vital that GPs are equipped to identify such problems accurately and early. I should note that given the relatively small sample size, our findings should be interpreted with a degree of caution. However, our findings compare favourably with other work reporting on GP attitudes to addressing youth mental health problems. Our sample included GPs mainly in the older age group, so further research should probably look at GPs in the younger age group and also those at the earlier stages of their career. To conclude, and finally, the key strategies to enhance the GP role to address youth mental health problems include further training for GPs in youth mental health, interagency collaboration across the healthcare services, and appropriate psychological services. I'd also just like to mention that further research is currently ongoing by 2 members of the Mental Health in Primary Care Research Group, my colleagues Davina Swan and John Baran. They both have posters here today. So Work in IT is looking at the development of a health finder tool for GPs to identify mental health problems and monitor them in general practice, and an evaluation of an educational intervention in youth mental health is also underway. So please feel free to find Davina and John. Yeah, there they are. If you want to find out more about this research. Finally, I'd like to thank the GPs who participated, the HRB for funding, and our colleagues for their help as well with the study. Thanks.